It looks like we have a good number of people here already. So yeah, I'll just go ahead and get started and say welcome everyone. My name is Janae Adams and I'm the host for today and also the founder of Black Women Comp Bio. This is the last Black and Comp Bio seminar that we'll have for today at this part of the year. We'll probably return sometime at the end of the summer or in the fall. So just wanted to thank you all for your time and in joining us into celebrating another amazing pathway into computational biology by actually one of our members, Dr. Belinda Akpa. So just thank you all for your time. We have another member here with us today, Amanda, who will go ahead and introduce our speaker to you all.
Thank you, Janae. Hi, everyone, and welcome to the Black Women in Computational Bio seminar. Today, we have Dr. Belinda Akpa, who is a senior scientist at Oak Ridge National Laboratory and a joint associate professor at the University of Tennessee in Knoxville. She now focuses her work on computational systems physiology. She uses numerical simulations to bridge length scales in integrated computational and experimental investigations with the aim of accelerating the translation of molecular and cellular level findings in her applications. Her current interests lie broadly within the realm of quantitative systems, physiology, encompassing pharmacology, toxicology, and plant biology. So she will be talking to us today about molecular dynamics in simulations of CURT2 interactions with an ensemble of cholesterol molecules. I just want to remind everyone again that today, if you have any questions, please put them in the chat or at the end of Dr. Akpa's talk. We will also give you privileges to unmute yourself and ask your questions. Thank you, Dr. Akpa, for coming. You may now have the mic.
Thank you very much, and thank you for that introduction. Let's hope that this works.
Looks good.
All right. So I'm happy to have the opportunity to speak to you all today. My understanding is that we might have an intro to computational biology class joining us today. And I know even within our BWC network, we span a wide range of interests. So I'm going to do my best to make this accessible to a broad audience. My research interest is really in using mathematical modeling as a supplement to the broader experimental toolkit. Using mathematical models to think about how we design experiments and develop targeted strategies to be able to accelerate biological discovery. And the project I'm going to talk about today, you're going to see there is no experimental data in this project. It is a purely computational project where the data that we're going to analyze and interrogate in order to learn something useful about physiology itself, that data will come from a simulation. And I'll talk you through why that is and what kind of simulation that is. But generally what we're trying to do is use the molecular modeling as a thinking space that can basically propose hypotheses that might be worthy of experimental interrogation. So the topic today is ion channel function, and in particular the function of a potassium channel. Ion channels are proteins that are embedded in cell membranes, and they regulate the movement of particular ions from the outer environment into the cell or vice versa. They regulate that ion flux by opening and closing in a very well-controlled fashion in order to achieve healthy function of the cell. And if there's a defect in that opening and closing function, then you get contributions of channel function actually to disease rather than maintenance of a healthy state. We're going to focus today on cardiovascular disease and the way in which dysfunction of ion channels actually can prevent blood vessels from being able to contract or dilate in response to blood flow in order to regulate blood pressure. And so in the setting of hyperlipidemia, which is the elevated triglycerides, elevated cholesterol in the bloodstream, you're actually going to have an example of ion channels responding to that context by changing their function in a way that impairs their ability to respond to blood flow. I'll talk a little bit more about what I mean by changing their function as we move forward. But this class of disease is known as a channelopathy because it's associated with misfunction or dysfunction of ion channels. And typically, when we think about channelopathy, we're talking about genetic disease. So mutations that change the nature of the protein structure, and the protein structure change leads to a change in gating function. There are a number of hormone disorders and neuropathologies that are in fact genetic channelopathies. The case we're talking about today is not a genetic channelopathy, it's called an acquired channelopathy. And this is when the protein structure itself is a normal protein structure, but something changes in the physiological context of the protein that impacts its ability to change its shape as would be needed in order to open or close the pore to regulate the flux of ions. The channel that's shown on the right here is called KIR. It's the 2.2 variant of this channel. KIR stands for potassium inwardly rectifying. Inwardly rectifying just means that it carries potassium channels primarily as an inward flow of ions from the outside of the cell into the cell. And what's interesting about this particular class of acquired channelopathy is it arises due to interactions with a component of the cell membrane that is a normal component of the cell membrane, which is cholesterol. So there are acquired channelopathies that arise due to exogenous species entering the body, like heavy metals or a drug or some other toxin interacting with the ion channel, impairing its ability to regulate ion flow. And in this case, what we're thinking about is the channel residing in its normal milieu, which is the lipid membrane. And that lipid membrane contains as one of its major constituents, cholesterol, under normal conditions. But when you start to get elevated levels of cholesterol in your system and that gets incorporated into the membrane, then you start to have dysfunction of this particular channel. So what is it about this change in the level of a normal component of the membrane that actually alters the function of the channel? If we could understand that, we would have a better appreciation of how disease associated with this condition arises. And of course, we ideally would love to be able to intervene with some kind of appropriate therapeutic. So how does cholesterol actually impact the function of this channel? Okay, I said there'd be no experimental data from my work, but here's some work from my collaborators' lab. So I got involved in this line of inquiry through interaction with Dr. Irena Levitan at the University of Illinois at Chicago College of Medicine. Her lab studies ion channel function and cellular biomechanics in the context of development of atherosclerosis. And so here we have a plot from a cell physiology experiment where they measured current density through an ion channel as a function of cholesterol concentration. And what you find is this would be the normal operating point. As you increase the level of cholesterol, the current through the channel actually falls. And so cholesterol is a negative regulator of KIR channel function. Let me get a laser pointer going here. There we go. So here's your normal operating point. If you increase cholesterol, you get a lower current through that channel. Actually, if you decrease cholesterol, you can get a higher current through that channel. So there's something that cholesterol is doing that regulates the ion flow. There are a few different ways that constituents of the lipid membrane in which this protein resides can actually impact its function. One way is simply by changing the mechanics of the environment in which the protein sits. And so here's an ion channel protein with the possibility of transit through a pore between its subunits. Here is represented the lipid bilayer making up the cell membrane. Cholesterol, when it's added to a lipid bilayer, has a tendency to make it increasingly rigid. And so you can imagine if you're a protein and you want to be able to change your shape in order to allow for the pore to either open or close, you're going to have to do that by working against the rigidity of the membrane in which you are situated. If the mechanics of the membrane changes, then the energetics of that transformation change to either make it harder or closer for the evolution to occur. So simply altering the mechanics of the cell membrane would be potentially enough to alter the function of the ion channel. Now, my colleague has done work over several years to show that this doesn't seem to be the mechanism by which cholesterol acts on these KIR channels, doing experiments like taking optical isomers of cholesterol. So cholesterol has chiral centers, and you can take isomers that are almost identical in structure except for what's happening around those chiral centers, add them into the cell membrane. They will impact the mechanics of the membrane, but the structure is different enough that you would expect that they wouldn't, for example, interact directly with the protein by binding to it in a way that alters its function. And what they saw was that just changing the mechanics of the membrane is not enough to actually alter the function of the channel. So it looks like, coupling that information with some other experiments and computational work that has been done over the years, there is some kind of direct interaction that's happening between cholesterol and the ion channel. So cholesterol is in the membrane, it migrates somehow to the channel. We think it's going to interact with the channel. The next question we want to ask is, where is it interacting with the channel? What is the binding site? And then what are the consequences to channel function of binding to that site? So if cholesterol binds to the channel, how is it getting there from the membrane? And given that it's naturally in the membrane, what is it about increasing the concentration of cholesterol in the membrane that suddenly starts to impact the function of the channel, whereas it did not under normal conditions? Those are the questions that we wanted to address. And taking a little diversion for a moment, when we do the work in my lab, we are often thinking about molecular phenomena. There's typically some kind of physiological observation that a collaborator has brought to us that they don't fully understand, but they have some ideas as to what might be contributing molecular mechanisms. And so we come in and we start thinking about doing various types of computational investigations that explore those molecular mechanisms. We do it computationally rather than experimentally, often because there isn't a good experimental tool to directly observe the molecular phenomena of interest, or perhaps there are tools, but you want to explore the possibilities and think about what is the optimal experiment to run in order to save yourself the effort of committing both time and resources to doing what might be more of a fishing expedition, exploration of the possible phenomena at play. In this case, it was true that trying to directly observe the interaction between cholesterol and the channel in something that is similar to a native environment within the membrane is difficult to do experimentally. And so we used a molecular simulation approach. So in this talk, I will go over what molecular simulation is and how that allows us to generate data describing what's happening in the membrane, what's happening with the protein, and what's happening with the interaction between cholesterol and the protein. I'll then talk about how we use network concepts to process that molecular simulation data and start to answer that question: is cholesterol interacting directly with the protein? If it is, where is it interacting with the protein? And we're also able to take that network concept and change its definitions in such a way that with the exact same data set, we could also start to ask, well, what is cholesterol doing to the protein once it's bound? How does the dynamic structure of the protein change that impacts its overall function? Ideally, however, what we would want to do is predict something with our computational investigation that is measurable in an experimental setting. So we usually want to get to the point where we can predict something we know is measurable, like the ion channel current, which I showed you on the previous slide, because that then allows you to compare an experiment to a computational prediction that we know is grounded in a particular set of molecular phenomena that we think might explain the physiological observation. And that allows us to validate our computational predictions, or gives us new information that allows us to go back through this loop and reassess what we think might be causally at play in that system. So we're going to do this part. We're going to get to the network analysis and what we learned from that. There's a natural break in the talk at that point, and depending on how we're doing for time, I might be able to show you this piece, which is how we took what we learned from the network analysis and what was happening to the structure of the protein, and we're able to create a dynamic Markov model simulation that allows you to extract a predicted current. So time for molecular dynamics 101 for those of you who aren't familiar with this type of simulation. We want to understand molecular motion, molecular interactions, and what you can do is you can take the structure of a molecule, even a molecule as complex as this Keir channel protein with its large number of atoms, and you can think of every single atom as a particle. And so you have these particles in space that interact with each other in various ways. If you have two atoms that are bound together, then they interact in such a way that they're not capable of moving far away from each other. They're not capable of moving too close to each other. They have a sort of natural bond length that they reside away from each other. And if they move away, then they'll experience a restoring force that brings them back together. If they move too close together, they experience a force that pushes them apart. And in fact, that should be very similar to what you've learned in a basic physics class about spring harmonic potentials. And that's actually the sort of equation that you would use in a molecular dynamics simulation to describe the behavior of two atoms that are bonded to each other. You can think, instead of being bonded to each other, what if these two particles are in each other's sort of sphere of influence and they're carrying an electrical charge? And so then they'll experience electrostatic interactions as they interact with each other through space. If they have like charges, they might repulse each other. If they have different charges, they might attract each other. And so you can use an electrostatic potential equation to describe that sort of sphere of influence through electrostatic charges. So there are other ways that these particles can interact through things like hydrogen bonding and other through space interactions. If you can describe all of those interactions with energy functionals, then those energy functionals actually give you a way of calculating the force that the two particles are exerting on each other. So the gradient of the potential is the force. Okay, so now with your 12,896 atoms, you can go pairwise and say, this atom, how are they interacting with each other? Calculate the force between them. Next pair of atoms, etc. And that allows you to calculate for every single particle what is the net force that it is experiencing from all other particles in the simulation. Now it's a matter of integrating Newton's laws. Force equals mass times acceleration. If you know the force on the particle, you can calculate its acceleration. If you have its acceleration, you can take a step forward in time and you can predict what happens to its velocity. If you have its velocity, you can take a step forward in time and you can actually predict what has happened to its position, how it has moved. Loop back to the beginning with all those new positions, recalculate all the forces, update the velocity, update the position, and so on, and repeat for however many time steps you need to reach the length of simulation you desire. Okay, so you have to do that now for all of these atoms doing these pairwise calculations. You need to keep track of each atom's position as a function of time, each atom's velocity, and the force that's exerted on each atom. So it's lots of calculations and lots of information that you're preserving. The time steps that you can take forward are fairly small because they're limited by the fastest movements that occur in the system. So you're looking at femtosecond size time steps, that's 10 to the minus 15. If you wanted to run just a nanosecond of simulation, that's already doing a billion iterations of this set of calculations. So these kinds of computational simulations are very computationally expensive, and that limits the size of the simulation as well as the time scale that you're actually able to simulate. 1 nanosecond is not approaching the time scale at which an ion channel actually opens and closes. So we're not actually going to be able to use this type of simulation, which is an atomistic simulation, to access the timescales of opening and closing. But we can do a little bit better than the order of, say, hundreds of nanoscales to a microsecond or more, if we adopt a different approach called coarse-grained molecular modeling. Conceptually, the exact same as what I showed you on the last slide. What we're doing is reducing the number of degrees of freedom, the complexity, the number of computations, by instead of representing every single atom as a particle, you actually group atoms together. So using the Martini coarse graining scheme, which is what we used here, that's typically 4 heavy atoms together. So heavy would be things like carbon, hydrogen, oxygen, nitrogen. We omit the hydrogen atoms. Group them together into an interaction site, and then you calculate the motions of those interaction sites. That's enough to make it possible for us to begin to simulate not just the protein with its nearly 13,000 atoms, but also to simulate the lipid bilayer with all of its phospholipid molecules and the sterile molecules that are in that interface. And we don't want to just represent one sterile molecule because we're interested in how the level of cholesterol impacts the function of the channel. Ideally, we want to be able to represent quite elevated concentrations of cholesterol in that system. And so we actually have here depicted, I believe this is the 30 mole percent cholesterol simulation. So that's 429 cholesterol molecules in a milieu that has over 1,000 lipid molecules, as well as the representation of our ion channel protein. Okay, so you do your simulation, what do you get? You get X, Y, and Z coordinates for every single particle for every time step throughout the molecular trajectory. With the coarse grain simulation, that time step is about 20 femtoseconds rather than 1 femtosecond. So we've gone up by an order of magnitude. You can look at all of that data. It's an awful lot of data. Won't mean a lot to you just looking at the numbers, but you can also make a visualization like the one that I'm showing on the left here, which represents those trajectories as a function of time. We have here the phospholipid head groups, the phosphate groups representing the top of the membrane, phospholipid phosphate groups representing the bottom of the membrane. There are lipid tail groups here representing the hydrophobic core of the membrane, and all I've done is I've hidden those so that you can clearly see where the channel lies. So with this image from the previous slide, all of these species are still present. You can just choose which ones you want to visualize and how. And so I'm looking at the protein, which is made up of 4 different distinct subunits, so they are each colored differently. And then I'm highlighting here a particular set of residues in red that are interacting with a single sterile molecule. Again, all 429 sterile molecules are in the simulation, but we're choosing to visualize only one. And that allows us to answer the question, does cholesterol bind to KIR? That looks like a binding event. We have a cholesterol molecule that is residing for some prolonged amount of time in contact with several residues of the system. You don't want to sit there and go through frame by frame to try and identify these binding events. Ideally, you want to do something numerical and quantitative to characterize those events, which residues are involved, how does cholesterol get there, et cetera. And so that's where we start doing some crunching of this data that has come out of our numerical simulation. And what I'd like to tell you is a story about how the simple way of conceptualizing what binding is that has worked in several other molecular simulation studies didn't work for us. And it led us to think carefully about what do we really think a binding event is? Is it simply contact between a ligand and the protein, or can we say something more sophisticated of the nature of that contact, and in fact use those relationships to extract from what is a very large amount of data, but with very few binding events, what is the nature of those rare events? Okay, so I told you we have this massive data set that has all these X, Y, and Z coordinates for all of these particles over time. And we ask ourselves, how am I going to find a binding event? Well, one way it's been done previously is to simply look for contact between the ligand and the protein. And so we have to define what we mean by contact quantitatively in terms of those X, Y, and Z coordinates. So you could say, well, if I'm in contact, I must be in close proximity. So let me set a threshold for that proximity. In this case, we use 6 angstroms, and calculate the distance between a particular protein residue and any sterile molecule in the system. Anytime I see that that distance falls below 6 angstroms, I'm going to count that as a contact. And so if you go frame by frame through the trajectory, count the contacts for each residue, and you would think if a residue is involved in this kind of a binding interaction, you'll have a high number of contacts, and so that would be something that you could define as a contact residue. That has worked really well in the past for this particular protein for a different ligand called PIP2. So PIP2 is a lipid that's actually relatively low in abundance in the membrane, and it interacts with this KIR protein via electrostatic interactions, which are stronger interactions than some of the other through space interactions. And so when PIP2 comes in contact with the membrane, with the protein, it does so in a very well-defined way, interacting with charged residues near the interface between the membrane and cytoplasmic domain. The protein itself, as I said, has 4 different subunits that come together to form the channel. And there'll be one of these binding sites for each subunit. And so when you process the data, you will find that on average, there are 4 PIP2 molecules in contact with the protein, 1 for each subunit, tightly bound, and they stay there for the duration of, say, a simulation that runs tens of microseconds. So we do the same analysis with cholesterol, and we find that it's not so simple in this case. Just calculating the number of cholesterol molecules that are in contact with the protein at each time point, so the number of molecules on the y-axis, time on the x-axis, we actually find that there are 5 sterile molecules per subunit in contact with cholesterol, not just 1 per subunit. Okay, that's interesting. We can plot a histogram, count how many contacts each residue experiences with sterile molecules over the course of the simulation, put the contact time on the y-axis, and identify the residue on the x-axis. We actually find that there's a large number of residues that seem to show that they have frequent contacts with the sterile molecules. And if we actually render now on a structure of the protein, those residues that seem to have, say, the top quartile of contact times from this histogram, we find that they're distributed all over the transmembrane domain of the protein. There doesn't seem to be a single well-defined binding site when you simply map frequency of contacts from a residue's perspective. So I'm saying I'm a residue, how many contacts of sterile do I experience over the course of the simulation? So this doesn't get us to identifying a binding site. Is it possible that there just isn't a well-defined binding site? Is it possible that there's just sort of a region of the protein that recognizes the abundance of cholesterol? We didn't stop there. We said, okay, well, there might still be a site. Maybe we're just not asking the right question of our data. So we thought about, what is the nature of a binding site? A binding site is not a residue in contact with a sterile molecule. It's a cohort of residues that sort of form a favorable slot for the ligand to interact with the protein. Residues concurrently in contact with the sterile molecule. So if you start to look at the different contact instances now from the cholesterol molecule's perspective and say, if I'm a cholesterol molecule, how many protein residues am I in contact with when I form a contact with the protein? We actually found that the vast majority, 95% of our contacts are short-lived and only involve a few residues. And so we have number of contact events on the y-axis, number of residues in the contact cohort on the x-axis. 95% of the interactions are with 3 residues or fewer. It is a rare event where we see contacts with a large number of residues, say 9 or more. And those are the contacts that look more like what we saw in the video on the previous slide. Those seem to be our binding events. We can take a step further and say, given the size of the residue cohort, how long does the contact last? And what we find is that it's only those contacts that involve large numbers of residues, 9 or more, that last on the order of microseconds. Our total simulation is about 20 microseconds in length, and you see that we have several interactions that last on the order of microsecond or more. Most of the contacts last 10 nanoseconds, hundreds of nanoseconds. And so what we can do is say, our binding events are not simply contacts. They're a particular type of contact. They're a contact that is long-lived and involves a large number of residues. So let's exclude all of the data that has the characteristic of these short contacts and continue with our sterile-centered perspective on asking what a binding site looks like. So now I'm going to add to that perspective and not just say that it's sterile-centered. I'm a sterile molecule, I'm interacting with different residues. There's a relationship between those residues too. If they're part of the same binding site, they should both be interacting with the same sterile molecule at the same time. So how do we build all of that relationship information into our analysis so that we can somehow identify what these binding sites look like from our data? This is where we decided to take a network perspective, because networks are very good at highlighting relationships. So for those of you who aren't familiar with network analysis, networks are actually fairly simple on the face of it. It's a type of graph that allows you to sort of organize relationships between agents of interest. Relationships of interest, agents of interest. Emphasizing that because it is up to you to choose the definition of a node, which is your agent of interest, and to choose the definition of an edge. What is the relationship that you're interested in? So in our pandemic moment, a node might be something like an individual in the community, and an edge might be representing the fact that that individual has had prolonged contact with another individual in the community. So for example, you can imagine two people who live together, like family members in a home, or students sharing a dorm room, might be connected by an edge because you know that they're going to spend prolonged amounts of time together. Those kinds of networks can be very useful in thinking about disease spread. Here's another kind of network that I pulled up yesterday, where I use a tool called Mention Map and plugged in my Twitter account, and it generates a network where the nodes are Twitter accounts and the edges represent whether or not my Twitter account, which is in the center here, has tweeted something that mentioned one of these other accounts. So you can see Black women in computational biology here. So I'm not a prolific tweeter, getting more active recently, but I certainly have more followers and I follow more people than are indicated in this network. The reason you don't see all of those is because of the definition of the edge. If we define the edge as tweets that contain a mention, that reduces the number of nodes that need to be in that network. Also, you might notice that these edges have different thicknesses. That's called the edge weight. And you can use the weight of the edge to indicate some other information about the relationship. And so for this Twitter network, the edge is actually indicating how many times my account has mentioned the other account. And so by defining nodes, defining the relationships you want to represent in edges, defining edge weight, you start to shape the nature of your network. And beyond just doing a graphical representation, you can actually start to do some quantitative analysis to learn from that network. How does this then apply to proteins and ligands? Okay, so we're conceptualizing a cohort as a group of residues interacting with a particular cholesterol molecule at the same time. So what we did was we created nodes, and each node represents a residue, and the number represents the residue number. Each edge represents whether or not the two residues make simultaneous contact with the sterile molecule. How we identified simultaneous contact was by frame, using an algorithm to say, in this frame, for this sterile molecule that we know has the properties of a binding contact, which residue is it in contact with? And so for, say, residue 89, as I go frame by frame through my molecular trajectory, I'm either going to have a 1 or a 0, representing am I in contact with that residue or not in contact with that residue. I would do the same for residue number 93, 171, 172, et cetera. All the residues in the protein, 1s and 0s. I can look at the pattern of 1s and 0s for each residue, and I can calculate a similarity metric. Because if they have very similar patterns, that suggests that they were concurrently in contact with the same molecule. And so where the nodes represent residues, the edges actually represent that similarity metric. How similar is their pattern of contact over the course of the simulation? That means that you won't see any nodes in this network if they never contact the protein. And you won't see an edge if two residues aren't concurrently in contact with the protein. The edge weight represents that similarity metric. So a heavy edge means that these two residues were frequently in concurrent contact with the sterile. The node size we use to represent how long that particular residue was in contact with the sterile molecule over the complete duration of the simulation. Finally, the node color here represents different clusters or modularity groups. What those are simply groups of residues that have more interactions or edges joining them together than they have edges joining them with other groups of residues outside their cluster. And so we use, I think it was Gephi in this case, but one of these network analysis software packages to actually distribute the nodes into their different neighborhoods. And we identified the neighborhood that had the prolonged contacts as the one that was most likely to be the binding site. Okay, so that's great. You do all of that, you get that information for one residue. Over the course of, say, a 20 microsecond simulation, and we ran 4 different instances of the simulation, we found 25 sterile molecules that had these kinds of prolonged contacts. We did the network analysis for all 25, and that means for each contact, we now have a characteristic set of features that is the list of residues involved in that binding event. They're not going to match exactly, but you might see some overlaps between the different residue lists, like this sterol interacted with residue 171, and this one did as well. But are they the same contact site or not? To answer that question, we actually did clustering. So this is k-means clustering, which is another way of grouping things according to similarity. And what we found is that 3 different clusters seem to emerge from our data. In this plot, we have the identity of the cholesterol binding event on one axis and the identity of cholesterol binding event on the other axis. And so if I look at event number 6 and I move up to event number 5 here, I see a high phi coefficient, that's similarity coefficient. That basically means that those 2 binding events consisted of residues that were very similar. They're probably the same binding site. Versus sterol number 6's binding event compared to sterol number 20, very different binding profiles, different residue lists. So those get assigned to a different cluster. So we get 3 clusters. Here, similar information presented, but here we have a dot that indicates for a particular pair of binding events, was that phi coefficient, the similarity coefficient that we evaluated, was it statistically significant or not? And so if it's statistically significant, then we indicate that with a dot on this plot. Again, we see 3 groups, but you might be asking yourself, there are a lot of statistically significant correlations in these areas here. Why am I calling these 3 binding events instead of, for example, 2 binding events? Well, if you take the centroid that was identified by the clustering analysis, so the centroid is, I looked at these events, I determined that these ones are all similar, and then I identify what is the characteristic set of residues that identify this particular cluster. That's the centroid. I go to my protein structure, which is represented with these ribbons here, and I use surface rendering here to highlight the residues that were identified as the centroid, the characteristic residues of the clusters. And so we have what I'm calling site 1 and site 2. They both occupy a space that is between alpha helices in the transmembrane region. This is between helices of 1 protein subunit. This is between helices of 2 adjacent protein subunits. This site here corresponds to this distinct binding cluster here. The other 2 clusters correspond to what I'm calling site 1 in this case. If you zoom in, what you find is that site 1 actually seems to define 2 distinct pockets, but those pockets share 2 residues in common. Moreover, when cholesterol interacts with either of these 2 sites, how stable that interaction is actually depends on whether the other site is occupied. So if only 1 cholesterol molecule is present, it can actually slide a bit between what I'm calling site 1A and site 1B. But if 2 cholesterol molecules are present, you get these 2 very well-defined distinct sites. And just to prove that those are in fact distinct sites, here's another rendering of the protein where I actually show a snapshot of the simulation where these cholesterol molecules are rendered in orange and all 3 sites are concurrently occupied. So what we identified with our numerical analysis via this network approach bears out when we go back to the simulation to look for the events that were characterized. Okay, that's great. We've got 3 binding sites. Rewind back to the phenomenon that we're interested in. Cholesterol is always in the membrane. It is a natural component of the membrane. It must be interacting with protein somehow under normal conditions, but when cholesterol levels are elevated, the protein starts to have defects in function. So what is it about cholesterol binding to these sites that might differ as a function of concentration? So we ran another set of simulations where we reduced the concentration of cholesterol by a factor of 2 so that we could check to see what the nature of the binding sites were. We found the same binding sites in the low concentration simulation, but the occupancy of the binding sites changes. So at the high concentration, it's not unusual to find all 3 binding sites occupied. And if we look at the fraction of the simulation time where we see the binding sites occupied, we find that all 3 binding sites are occupied for at least 50% of the simulation at the high cholesterol concentration. At the lower cholesterol concentration, we see binding site occupancy for site 1A and site 2, not so much for site 1B. And so site 1A is always occupied, but it looks like there is a distinct set of sites that become occupied only at higher concentrations. Maybe site 1A needs to be occupied for normal function of the channel, and then 1B and site 2 might be responsible for the deleterious effects on the function of the channel. Everything I've showed you so far was based on the open structure of the channel. And so we repeated the calculations, did another set of simulations, starting with the closed conformation of the channel, looked for the binding sites to see whether in fact we found the same binding sites as in the open configuration. And so that's that analysis here. We again found 3 clusters. The clusters are again located between alpha helices of the transmembrane domain, either between alpha helices of one subunit or between neighboring subunits. But we found some subtle differences in the identity of the binding site. Namely, although site 1B was the same in both cases, the lower site in either this groove or in this groove actually was displaced downwards in the structure of the protein. It was a distinct binding site that we found, but still in the same groove. So that's interesting. And it leads us to start to posit what might be happening in the transition between the open state to the closed state with this particular channel. We find that cholesterol initially binds, or we posit that cholesterol may initially bind to the channel in its open state. That binding is driven partly by interactions between cholesterol, which has a hydroxyl group at its head, and polar residues on the protein that actually reside in these binding sites. Those were the ones that were actually highlighted in a different color here. So you have hydrogen bonding interactions and polar interactions that dictate cholesterol entering those binding sites. Binding to the open channel, we're suggesting, may help disengage the gating domains that have to be disengaged for the channel to close. So there's a cytoplasmic domain of the protein, there's a transmembrane domain of the protein, and as you move between open and closed, the cytoplasmic domain sort of twists and either engages or disengages to change the size of the pore opening and the chemical character of the pore opening, so that regulates flux of the ion channel, of the potassium ions. So here's an idea. Cholesterol binds to the open state. Cholesterol changes the structure of the protein in such a way that the gating domains are disengaged and makes it easier for the channel to close. And then, because cholesterol is already in those grooves, it can slip downwards into these new binding sites that we identified for the closed state and perhaps stabilizes the channel in that closed conformation. That would certainly inhibit the opening of the channel, which would lead to a reduction in current that travels through the channel. At this point, this is a mechanistic hypothesis. We haven't explored anything dynamic. So a summary here of what we found through this part of the investigation. We wanted to understand how cholesterol moves from the membrane to the protein, and when it gets there, how does it interact with the protein, and why does that change when the cholesterol concentration changes? What we found was, yes, cholesterol interacts with the protein and it binds to particular binding sites. What I haven't showed you, which you would see from a dynamic visualization, is that cholesterol doesn't spend a whole lot of time in those binding sites relative to, say, the time scale of channel conformation changes for gating of the channel. Our simulation was about 20 microseconds in duration. Those interactions were maybe 1 to 10 microseconds in duration. So cholesterol goes into a binding site, cholesterol can pop out of the binding site, but if there's enough cholesterol in the membrane, another cholesterol molecule can come and pop in. And so from the protein's perspective, its binding site is always occupied simply because there's enough cholesterol in the membrane to drive that occupancy, even though the actual binding event is not terribly long-lived, and so probably not a very high affinity binding event. So the mediation of the protein function may not be dictated by sort of a high affinity protein lock and key kind of protein ligand lock and key kind of interaction, but rather by a systems phenomenon that depends on how much cholesterol is present in the membrane. Okay, looks like we're at 1:47, so I will touch on this and then I'll take questions. I put forward this hypothesis, which is really related to protein function, and nothing I've shown you so far tells you anything about protein function. I want to just highlight the fact that with the exact same data set, we can go back to this concept of network analysis, and if we change how we're defining the nodes and the edges, that same data set can start to tell us about how the protein structure changes as a consequence of cholesterol binding. And so the new network that we're going to define is going to have each node be a protein residue. And each edge is going to indicate whether or not the residues are in close proximity to each other. That proximity might be because they're bound together covalently, or it might be because in the 3D structure of the protein, the folds bring those residues quite close together.
Okay, yeah, sorry.
Once we do that, and we take all the different simulations that we've run, open state, closed state, high cholesterol, low cholesterol, we look at all of those simulations and say, across all of these conditions, which contacts persist, which residues are always in contact with each other. We preserve those edges that represent 90% of the time these two residues are in contact in any of these conditions. Our thought being that if you have that sort of conserved contact between residues, then they must be part of a domain that moves coherently as the protein evolves, like a gating domain. So we have our nodes, we have our edges, we do our sort of neighborhood clustering to pull together groups that are more interconnected than they are connected to other parts of the network. And that allows us to actually segregate the protein structure into what we are thinking of are coherent domains of motion. And so we find 8 different domains, and it just so happens that the domains we identified in this network fashion seem to tie up with what the literature reveals to be functional domains of the Keir channel. We can then take each of those domains and say, okay, I can add up all the ways that one domain can interact with another domain, looking at residue-residue contacts. And I can say, comparing, say, the low concentration cholesterol case to the high concentration cholesterol case, is the difference in contact between these two domains changing such that they're in aggregate losing contact or gaining contact? How big of a change is present? And so again, each of these nodes is representing something about the structure of the protein. They're representing the different domains that we identified. And then the edges are actually showing you how much of a change there is in contact between the domains. Red means there's a loss of contact, and the heavier the line is, the bigger the loss of contact. And so we're predicting here that what's happening to the dynamic structure of the protein is that we are really decoupling the cytoplasmic domain from the transmembrane domain, and in particular, decoupling this G-loop region from some of the other components of the cytoplasmic domain. So that's starting to get to the idea that indeed the cholesterol molecule binding seems to be driving a change that drives the protein structure into this decoupled conformation, which is more like a closed state. This is for the closed state of the protein. You can do the same analysis for the open state, and it leads to a similar conclusion. So I'm going to stop there. Thank you for your attention. Happy to take questions. Thank the people who contributed to the work. Again, my colleague, Dr. Reina Levitan at UIC, whose group focuses on this kind of ion channel functional study in the context of cardiovascular disease. My PhD student, Nicholas Barbera, who's responsible for all of the molecular simulation. We worked together to do the network analysis. He's now a postdoc at the University of Virginia. And what I didn't get to talk to you about, which is taking this information about dynamics and pushing it even further to see if we can use different clustering and featurization approaches to learn from the simulation data what kinds of states arise out of the change in dynamics of the protein as a consequence of cholesterol binding, we were able to use those states to actually learn enough to develop a dynamic model to predict current through the channel. That was the work of my PhD student, David Flaherty. So thank you.
Thank you so much, Dr. Akpa. Does anyone have any questions? Okay, so I have one quick question. I missed something. I just wanted to know if the concentration of the iron affect the duration of how long the sterile binds to the pocket. That was my first question. That you increase or decrease the concentration, could the sterile bind for a longer period in the pocket?
Did you ask about the concentration of the ions or the concentration of the sterile? Sorry, I didn't quite hear it.
The iron.
That's an interesting question. We are not actually modeling ion transport in this case. And so it's not something that we've considered, but it's an interesting question that the ion concentration probably has a role in triggering opening and closing of the channel. So whether there's an interaction somehow between iron concentration and sterile concentration, that I don't know. We haven't considered it.
Thank you.
Mm-hmm.
I'm just looking for more questions. I don't see any. I did want to ask you something. Accessing your data. Earlier you mentioned that you have received data from a collaborator. Are there any other public databases that you can access the data that you run your simulations on?
So the data that we ran our analysis on is all from a molecular simulation that we did. None of our data actually comes from our collaborator. Where we engaged in this collaboration was that there was a problem or a challenge in terms of understanding what are the molecular scale phenomena that drive this important physiological event. And so then we go to the computational space and say, okay, let me simulate these molecular phenomena that are difficult to observe directly. Let me see what my simulations tell me might be at play. Let me predict then back at the physiological level what my simulated molecular mechanisms would suggest you should see at the physiological level when you, for example, mutate the protein, or when you know, as your previous question, if you think that ion channel concentration has a role and that's something that you can simulate at the molecular level, you can say, okay, I think the ion channel concentration does this as it interplays with cholesterol concentration. Now I can predict the current as a consequence. Please go do the experiment that has these conditions to see if my simulation is right, because if it is, then it suggests that what I predicted at the molecular level is valid. So yes, the work I presented here, all of the data comes from molecular simulation that we did.
Okay, thank you. So that was actually going to go into my next question, which kind of answered itself, because I was going to ask, do you from your simulations and go and run experiments themselves, and how difficult they are to run?
Yeah, so that's why all of my work tends to be in close collaboration with an experimental collaborator, is that I'm very interested in informing what you do experimentally. So I should say my PhD was not computational and it was not biological. I was trained as an experimentalist, and I started dabbling in models as a way of designing experimental protocols and trying to interpret my data. And I still take that sort of approach to my computational work now, which is how do I help experimentalists be very targeted about their hypothesis formulation and kinds of experiments that they proceed to do.
Awesome. Then I do have a couple more questions. The first question is from Brooke LaFlame and I'm sorry, Wade Davis. How do you sample your simulation space?
Wade, would you care to expand on that?
Yeah, I just didn't know in your last slide if it was maybe you didn't need to sample because you showed like high, low, and then I can't remember. It was almost like a 2x2, and so maybe you just did all four. But I didn't know in some situations you might have a lot of knobs to turn, and maybe you know, almost an infinite number of possible combinations. And so I didn't know if you kind of put distributions on some of the knobs you turn. Otherwise, you could just maybe keep trying so many possibilities, I would guess.
Right. So in our case, what we did was we aimed for cholesterol concentrations that, you know, with rough calculations, I think that our simulations were sub-physiological, normal physiological, and supraphysiological. So above and below the normal operating point.
Mm-hmm.
And then we looked at the open state and the closed state. We can't access a timescale long enough to actually see the evolution between open and closed. And I didn't mention that these coarse grain simulations, because you lose data when you coarse grain and you're no longer representing all the atoms, you have to do things like apply elastic mesh networks to help preserve the secondary and tertiary structure of the protein. And so that doesn't allow it to evolve between open and closed state, even if we could access those timescales. And so, yes, we have to sort of sample from the perspective of we can do open, we can do closed,
yeah.
And we can do different levels of cholesterol. And then the part that I didn't have time to talk about today is saying, well, now if I explore the open and the closed configuration to see if there are different substates that arise as a function of the cholesterol concentration, can I turn that into something that allows me to predict what might be the transition between open and closed, which is what you need to do to be able to predict currents.
Oh, I love to talk. Thank you.
Thank you for the question. Looks like we got a few more here.
Yeah, Taylor actually had a question as well. In the first network that you mentioned, did you notice a relationship between the density of your clusters and your definition of binding?
Taylor, do you want to expand on that? By density, do you mean the fact that the clusters seem to be quite close together and in fact shared some residues?
Yes, exactly.
So relationship between the density of the clusters and our definition of binding. I guess that gets at part of what I alluded to with why our analysis seemed to show some correlations between two of the clusters. So, you know, is it binding in the same sense if I have a cholesterol molecule plug into that top site in the groove? And as the simulation runs, if there isn't another cholesterol molecule in the bottom site, there's the ability for it to sort of slip up and down a little bit. So what is the binding site? I think the reason they segregate into two well-defined binding sites in our analysis is because we sampled enough events where both sites were occupied, and then you don't get that sliding back and forth. So that's an interesting question is, you know, how do you define a binding site when you have maybe that ability to slide back and forth?
Great, thank you so much.
Mm-hmm.
Okay, then we have another question from Brooke, and it's, for publications, do you feel experimental validations of molecular dynamic simulations is required?
Do I feel, or what has my experience been?
I think I'm looking more for your opinion because, so I'm an editor and yeah, we get a mix of opinions.
I don't think that experimental validation should be required. I mean, it's always nice if you can get there, but I do think that that holds back the ability of the field collectively to sort of build on this idea of models allow you to think through what prior observations might suggest as possibilities, which then can lead experimental interrogations in a new direction. It's great when I have a close experimental collaborator for us to be able to produce the computational predictions and then follow up with the experiments and have like a very comprehensive publication. But sometimes there are interesting problems that a computational scientist wants to go after where they can make real contributions in terms of thinking about the possibilities. That itself has value for somebody else to pick up and go run with. That should be published, in my opinion.
Thank you.
Dr. Akpa, do you have like time for 2 more questions?
I do, yes.
Okay. So the next one is from Anna Ritz. She asked, do you think there is any possible signal in those short-lived cholesterol contexts with 1 to 3 residues, or is it just noise?
Interesting question. So we spent a lot of time trying to figure out what was going on with all of those contacts and whether they were interesting or not. And one of the questions that we played with a little bit, which I didn't talk about, was how exactly does the cholesterol molecule get to those deep binding pockets? And we have seen some examples in our simulations where it looks like there might be engagement with some of those smaller groups of residues nearer to the top of the channel that helps guide the cholesterol down into the pockets. And so there might be a functional reason for those contacts to exist. And when we think about cholesterol in the membrane, our simulations currently have it evenly distributed in both leaflets, but our collaborator with others at the University of Illinois have looked at the distribution of cholesterol in the leaflets of a cell membrane and found that there's an asymmetric distribution. And I believe the higher concentration is in the outer leaflet. And so if our binding sites tend to be in the lower leaflet, there's that question of how does cholesterol get from one place to the other?
Thanks.
Thank you for the question.
And one final question. Oh, I just missed that. Oh, yeah. Beautiful figures and MD movie. Please, what software was used to create the figures?
Yeah, my PhD student, now postdoc in another lab, did some beautiful work in involving how we represent these proteins over time. That was VMD is what we use to make all of the figures and movies.
Okay, I think we have one last question, and then we'll close the chat for questions. So it said such techniques can be applied to intercellular loop of cytoplasmic tails of protein with other molecules. Oh, I think it was just a comment and not a question.
No, that was a question. I wanted to know if you could, if the Malay is too heavy to look at the intercellular loop or the cytoplasmic tail of a channel protein.
And when you say look at, so we are simulating the whole protein.
Yes.
A lot of times I only depict the transmembrane domain because we were looking for the interactions in the transmembrane domain. But the work that we're going on to do, which is being written up at the moment, also looks at, well, if you have this engagement in the transmembrane domain, what happens to the cytoplasmic domain? Are there responses there that are communicated through the structure of the protein? Prior work by my colleague has shown that there are cholesterol sensitive effects in the cytoplasmic domain, but clearly cholesterol isn't interacting directly with that domain. And so it seems like what's important is not so much what cholesterol is doing to the residues in the transmembrane domain, but how what it does to that part of the protein alters the structure and function in the cytoplasmic domain as well. And so, you know, with the same data set, this is all the same set of simulations, changing how we define the edges, changes how we define the nodes, change what we're asking of the data set, process it through different learning approaches, we're able to start to ask questions about the cytoplasmic domains and other parts of the protein. Does that answer your question?
Thank you. If it's okay, I'm going to get in touch with you because I'm looking at a membrane channel where you change the cytoplasmic tail and that closes the molecule or closes the pore or opens the pore depending on what interacts with the cytoplasmic tail. So I have your telephone number from the email, from the internet, and I also have your cell phone number, and I'm Sandra Murray and I'll be in touch.
Okay, I should say that the internet still thinks I work at NC State University. Sorry, you're muted.
Yeah, that's what I have, unfortunately. So what should I have? Not that it shouldn't, your telephone numbers doesn't start with 919 anymore?
No, it doesn't.
Okay, then I have a problem.
I will pop.
My name is Sandra Murray, and I'm going to put my email address in the chat.
I'm popping my personal email address in here as well. So that goes for anyone who wants to reach out. You can always reach out to me at my personal email address. And I have yours, smurray@pitt.
Yes.
So happy to answer anything else if there are any more questions about me personally, my path, or the talk.
I guess one last probe for the students that were here today, how, I guess, would you position yourself within the computational biology landscape, since this is more of systems bio? for people looking to get into comp bio, how would you differentiate if you do, and how does your background and kind of your education speak to those strengths?
Yeah, good question. Like I said, my PhD wasn't in this area. I didn't start out with my undergraduate degree thinking I'd be doing work like this. I'm a chemical engineer by training. I've worked both in chemical engineering departments and in a college of veterinary medicine. And now I'm at a national laboratory and affiliated again with a chemical engineering department. I love chemical engineering. I kind of picked it because I didn't want to have to choose which science to pursue, and chemical engineering is everything: math, physics, chemistry, biology, computing, and I think that it's made me very versatile. So I was able to ride some challenges that arose in my career and sort of redirect my research as necessary, from a PhD that was actually in experimental magnetic resonance to now running a group that does computational systems physiology, but with a mindset that thinks about what realistically you can achieve with experiments. You know, what are the strengths and limitations of both experiments and computational approaches, and how do you sort of merge the two so that you leverage the strengths of both to accelerate discovery. I think you do what excites you, and especially early in your education, you won't always know when something you have the opportunity to learn is going to be useful to you. You don't have to know at that early stage. Keep it in the back of your mind, put it in your back pocket. I've been astounded by how many things that I learned early in my education I'm now applying in my professional career. And I encourage students, yes, have an idea now what you're aiming for, but keep an eye open to other opportunities that arise to learn additional skills. It's that level of versatility and willingness to always be learning something new that is going to support you as you move forward in this wonderfully interdisciplinary field of computational biology. So I still say I'm a chemical engineer. I happen to be one who works in computational biology. I bring everything that I learned through that engineering education into the work that I do now.
Awesome. Thank you so much for the amazing talk. Everyone loved it. And thank you, Amanda, for navigating us through the discussion today. For those that want to learn more about us, please just visit blackwomencompbio.org. We're happy to interact and engage and continue having and creating spaces like this for people to talk about their science and build community. So thanks again. We look forward to hosting you all in the future. Have a good day.
Thanks, everyone. Bye.
Thank you.
Everyone, bye.